coevolution of humans and their gut microbiomes has been a dynamic process, driven primarily by major dietary shifts from ancient plant-based diets to meat consumption and cooking, and finally to modern industrialized diets. Each transition profoundly shaped the composition and function of the microbial communities, often in a mutually beneficial, though sometimes mismatched way. Key evolutionary transitions and microbial adaptations. Ancient plant-based diets. Early hominids primarily consumed a diet rich in fruits, leaves, and tough plant materials. Their digestive systems, like those of modern great apes, were larger and optimized for microbial fermentation of vast amounts of fiber. The associated microbiomes were rich in fiber-degrading bacteria that produced short-chain fatty acids, SCFAs, crucial energy sources for the host. Increased carnivory and cooking. The shift to consuming more meat and the control of fire for cooking represented a major evolutionary turning point. Host adaptations. Cooking made food more digestible and increased net energy gain, which supported the development of larger brains, but led to a reduction in gut volume and smaller teeth in humans compared to our ancestors. Microbiome adaptations. The gut microbiome adapted to a higher protein, lower fiber diet. This was selected for different bacteria and functions, though humans retained generalist omnivorous traits. Agricultural revolution. The transition to agriculture introduced new staple crops, grains, tubers, and animal domestication, leading to a more consistent but less diverse diet in some respects. This change further shaped the microbiome, selecting for species capable of breaking down specific plant starches, as evidenced by genetic differences across populations with a long history of agriculture. Modern industrialized diet. The recent rapid shift within the last 100 to 200 years to a Western diet, characterized by highly processed foods, high fat, sugar, animal protein, and low fiber, has created an evolutionary mismatch. Microbiome changes. This diet is associated with a significant decrease in overall gut microbial diversity, a reduction in beneficial SCFA-producing bacteria like Prevotella and Faecalibacterium prausnitzii, and an increase in potentially pro-inflammatory microbes. Health implications. This modern dysbiosis, microbial imbalance, is strongly correlated with an increased incidence of chronic inflammatory diseases, including obesity, type 2 diabetes, cardiovascular disease, and inflammatory bowel diseases. In essence, the human gut microbiome demonstrates remarkable short-term plasticity in adapting to new diets, but the sustained, low-fiber, high-fat nature of the modern diet has caused a significant and potentially harmful divergence from the ancestral microbial balance. We need to improve our microbiome for a healthy gut and a healthy life.